by UMass Association. And we have a very interesting topic today, that is gene regulation and neurological disorders. So in a cell, gene expression is an important aspect of biological functions. Gene expression takes place by central dogma, that is DNA to RNA and RNA to proteins. But every gene do not need to express all the time. So in a healthy cell, to conserve the energy and to maintain homeostasis, gene regulation takes place. However, malfunction of these gene regulation part can lead to various disorders, and in brain cells, it is called neurological disorders. So our two speakers today, Meena Naz Anadolu and Yedwan Oscar, will be presenting in detail that what are these neurological disorders. They'll be talking about autism spectrum disorders, that is ASD, such as Fragile X syndrome and newly found Argonaute syndrome. So this is for the information that this event will be recorded and will be available at Umpa YouTube channel. You can recommend to your colleagues and students to these seminars. We have a previous seminars and the recent one on our YouTube channel. And nowadays we are quite active on our social media handles, so you can follow us on Facebook, Twitter, and Instagram as well. This is a disclaimer saying that the views presented here do not belong to UMass Chan Medical School, but they belong to solely to the presenters. And if you have any questions during the talk, you can type in chat box and we'll try to accommodate at the end of the talk. And also try to participate in the survey at the end so that we can make Science Cafe even better. And you can also suggest the topics you would like to hear in future about. Also, if you want to be the part of our organizing team, you can email us, or if you want to share your research with us, you can email us at umpa@umassmed.edu. So with that, I would like to invite our first speaker, that Meena Naz Anadolu. You can contact her on her email if you want to know more about her research. So Meena is a postdoctoral researcher in program in molecular medicine at UMass Chan Medical School. She received her Bachelor of Science in Biology and Neuroscience from McGill University, Montreal, Canada. She did her PhD in Neuroscience at Sosian's lab at McGill University and studying RNA granules and translational control in neurodevelopment. Currently at the Richter lab at UMass Chan, Meena is exploring how ribosomes can stalled and unstalled on mRNA implicated in Fragile X syndrome, a type of autism spectrum disorder, that is ASD. Meena is originally from Ankara, Turkey, and she's passionate about singing and learning how to surf. So thank you, Meena, for being our speaker today and sharing your research with us. And the floor is yours. Please go ahead.
Thank you so much, Rishi, for that wonderful introduction. I'm just going to share my screen with you. My slides? Yes? All right, wonderful, because I cannot see you. So I'm very excited to be here today and talk to you about the regulation of messenger RNAs in Fragile X syndrome. And thank you so much to the organizers for inviting me for a talk today. My name is Meena, and I'm a postdoc in Dr. Joel Richter's lab. And I thought the best place to start would be from the DNA. And I think my colleagues have a poll question for you if you're interested in participating. DNA is the source code, the manual life of all. The DNA in our cells encodes all of the components of a living organism. And it is essentially made up of these letters, which we call nucleotides. And it is coiled up in the form of chromosome and stored in every single one of our cells. And what I like to compare this to is a library with many, many books on the shelves where all of this information is stored. So if you partook in our little poll just now and you answered 99%, 99%, 99.9% of our genes are actually, our DNA is the same between different individuals. And it's incredible because only 0.1% accounts for the differences that we have. And it's amazing that we are more similar than we are different. My slideshow is not advancing. Okay, here we go. So these very small differences can have a big impact in our physiology and what we look like, our hair color, our eye color, our height, skin color, body type, and whether we're able to tolerate spicy food or whether we love or hate the taste of cilantro. And one example here is a point mutation in which one of the nucleotides in our DNA is changed from a T to a C. And even a small difference like this in this vast library can make us unique. Now, mutations in our DNA can also impact how our DNA is expressed. And essentially, our DNA is transcribed into a messenger mRNA copy. And how I'd like to describe this is you take one of the books from the shelves in the library and you take it over to the copier and make a photocopy. And that photocopy is essentially the messenger RNA. And then that photocopy, that mRNA gets translated into the language of polypeptides, which are the proteins that encode all of the components that we need in our bodies. And it's just like taking that photocopy that you just made at the copier and going to Google Translate and translating it into a language that you can understand. And through this process and how DNA expression is regulated, the same DNA in each cell can be transformed into different enzymes with different functions. So the same DNA can result in proteins in epithelial cells that are different than nervous cells, that are different than blood cells. And so the DNA expression and regulation of this process is extremely important. The reason for this is that mutations can also cause neurodevelopmental disorders like Fragile X syndrome. And Fragile X syndrome is the most commonly inherited cause of autism and intellectual disability in the world. And it can be diagnosed with a blood test because it is hereditary and it runs in families. It affects both males and females and can skip generations. Although it has more severe impact on males because males only have one X chromosome. So if they get the mutant copy of this gene, then they don't have a backup copy. And often we see in Fragile X individuals these symptoms of intellectual disability, attention deficit and hyperactivity, anxiety and unstable mood, repetitive movements like hand flapping or not wanting to make eye contact, hypersensitivity to loud noises or bright lights, as well as speech delay and difficulty with expressive language. So these individuals understand language perfectly fine, but they struggle to communicate with the outside world. And about 10 or 20% of them can experience seizures. Now, it's important to note the difference between Fragile X syndrome and autism because Fragile X syndrome is diagnosed with a blood test and autism is diagnosed by a checklist of behaviors. And about 46% of males and 16% of females with Fragile X also are diagnosed with autism. And I just want to remind you that autism is on a spectrum and each person with autism is different. And individuals with autism can live very fulfilling and meaningful lives. And oftentimes they struggle in school because they may not do well in traditional learning environments and might need some additional support. But in the end, we must love and accept each other's differences because we are 99.9% the same after all. Now, Fragile X is caused by a mutation on this FMR1 gene that's located on the fragile site of the X chromosome. And what's special about this gene is that it contains this region that has this trinucleotide repeats. So this CGG nucleotides repeats many, many times. And if you have less than 55 repeats, then congratulations, you're making FMR1 mRNA, and your FMR1 mRNA is translated into functional FMRP protein, which makes you a neurotypical individual. However, if you have between 55 to 200 of these CGG repeats, then you might produce some FMR1 mRNA, but maybe not a lot of FMRP protein. And this could lead to some disorders such as ovarian insufficiency, tremor ataxia syndrome, and neuropsychiatric disorder affiliated with Fragile X. And individuals who have the full mutation often have more than 200 of these CGG repeats, which causes this gene to be hypermethylated and silenced, meaning you cannot transcribe or copy any mRNA, and you don't produce any FMRP protein. So you become an individual with Fragile X syndrome. Now, FMR1 gene is encoding this protein called FMRP, which stands for Fragile X messenger ribonuclear protein. And we know that FMRP binds mRNAs to control both their transport and translation in the cell. And we know that the loss of FMRP causes excessive amounts of translation to happen. So too many proteins are being made in the wrong places, in the wrong way. Send help. And we still do not fully understand the mechanism of how FMRP regulates mRNAs. So my job in the Richter lab is to look at these mRNAs and understand how FMRP interacts with them and what they have in common so that we can understand the mechanism of FMRP regulation. So here are some of the information that we already know and my scientific questions. So we know that FMRP binds mRNAs to determine their fate. And oftentimes when FMRP binds an mRNA, it causes translational repression. And when you don't have FMRP, all of these mRNAs are excessively translated and activated. So what do these mRNAs that are controlled by FMRP have in common? And what are their differences from other mRNAs that are not targeted by FMRP? We also know that FMRP represses translation by stalling ribosomes. So the molecular machine in our cells that is responsible for reading the mRNA and actually doing the translation to polypeptides is called a ribosome. And the ribosome reads, translates the mRNA, and through the help of some chaperone proteins, helps fold and wrap the proteins into functional enzymes. And we don't really understand still, how does FMRP interfere in this process? And I'm particularly interested in looking at polysomes that are regulated by FMRP. So poly means many, so many ribosomes can be loaded onto a single mRNA transcript and read it and translate it at the same time. And so these ribosomes or these polysomes can cause the production of a lot of proteins from only a single copy of mRNA. And if we take a step back, what our lab, in the Richter lab, is most interested in is how can we restore regulation? How can we regain control of mRNA translation in the absence of FMRP? And how can we help individuals with Fragile X syndrome? So the Richter lab is very interested in discovering therapies for Fragile X. Now, what do I do? What I'm doing right now is I am trying to capture these FMRP mRNA ribosome complexes using a technique called polysome profiling. And I essentially have two samples. One sample being a control, let's say a typically developing individual, a mouse, or some cells that produce FMRP normally. And to compare that to a mouse or cells that do not make FMRP. So we have removed FMRP from these samples or from these animals or cells to kind of replicate what's happening in Fragile X individuals. And what I do with this tissue is that I homogenize it and I load it onto a sucrose gradient. What it is essentially is a sugar solution that is about 20% sugar on the top of the tube and 50% sugar on the bottom of the tube. And we load these tubes into a centrifuge and spin them really fast. And because ribosomes are heavy, they tend to organize themselves in the order of weight through the sucrose gradient. And in the end, we have a little graph that looks like this, where we have neatly organized ribosomes from single ribosomes to light, medium, and heavy polysomes, depending on how many ribosomes are on the mRNAs. And then, so this is a sample trace that I have. And here on the x-axis, we have time in minutes, and on the y-axis, we have UV absorbance. Because ribosomes are dense and heavy, they absorb a lot of UV. So that's kind of how we detect them in our sample. And these peaks here that you can see in the center are the polysomes that I'm most interested in. And on the bottom is a Western blot, basically looking at the presence of FMRP protein. And as you can see with these black bands that are concentrated in this middle region where the light and medium polysomes are, these are the mRNAs and ribosome complexes that I'm most interested in. So I take these guys and extract the mRNA so that I can sequence them and see if these mRNAs are special. So basically, after I capture these polysomal mRNAs, what we do is we transform the mRNA back into DNA, because DNA that is double-stranded and is way more stable, we put it through a sequencer, and then we can understand, okay, what is the nucleotide sequence of these mRNAs and what is special about them? Are there specific regions where ribosomes stall? And is there anything important about these regions, like a specific motif or an order of nucleotides that this FMRP protein can recognize and bind to? So all of the mRNAs that are maybe controlled by FMRP could have similar features. And so that's one of the things that I'm most interested in, is to identify these features that these mRNAs have in common, and also maybe understand where the ribosomes might be stalling so that we can get a clue on the mechanism. Now, my project is only one of many projects in the Richter lab that is interested in Fragile X syndrome and FMRP. And I just want to give you a couple examples in case you're more interested in therapy. So one of our assistant professors, Dr. Sneha Shah, just published this paper, and I encourage you to take a look at it. And it's a antisense oligo-based therapy for Fragile X syndrome. It's very interesting. And my other colleague, Dr. Suna Jung, who recently became an instructor here at UMass Chan, is also working on FMRP and its role in splicing, because it turns out that some of these mRNAs that FMRP is regulating are encoding splicing factors. And it just shows that FMRP is such a master regulator of the cell's proteome. Also, I have another colleague in the lab who's working on something different, on CPB and inflammatory immune response in microglia. So if you're curious about the projects in our lab, you can also reach out to my supervisor, Dr. Joel Richter. His email address is here. So I just am running out of time, so I want to end by encouraging you to get involved in this kind of research. And I would like to ask you to connect with foundations like FRAXA that fund our research and other foundations, because they can provide a lot of resources and information on these disorders if you want to learn more. And you can also learn about the projects that they're actively funding that we're doing in our labs. Also, I want to encourage you to talk with family, friends, your teachers, medical professionals, so that we can increase awareness about neurodevelopmental disorders. And also, oftentimes when you go to the doctor to get a regular checkup, you are asked to give consent on whether you'd like to provide some samples, like donating cells, blood tissue, cerebrospinal fluid to scientific studies. And we rely heavily on those kinds of tissues, and we're always in need of typically developing healthy individuals to compare to individuals that have Fragile X syndrome. So I would like to encourage you to do that. And also, help become part of the solution by overcoming the stigma and be kind to neurodiverse individuals, because we are very similar after all. So if you have questions or would like to connect with me, please send me an email. And for those of you who are more interested in diving into the science, I'm giving a talk next month, April 4th at 3 p.m. at Biotech 2. And I'm going to be talking more in detail about my polysome profiling, and we'd be able to discuss in more detail the science. And thank you so much. And I'll be available to take questions now.
Thank you, Mina, for such a great talk. So if you have any questions, please type them in the chat box, and I will ask them on your behalf. And perhaps I can get us started with a question. So you mentioned that there is various types of treatments that are currently in development for Fragile X syndrome. Can you mention a few that are either being worked on in your lab or in other labs? Are there any gene therapies or small molecule inhibitors that are currently in the pipeline for Fragile X syndrome?
Absolutely. So there are a lot of labs that are working on Fragile X syndrome, particularly because the problem seems to be simple, right? There's a mutation that we can maybe correct with these new tools that we have in DNA editing, like CRISPR, Cas9, etc. So there are a lot of attempts and studies focusing on cutting and removing the CGG repeats or replacing the silent genes, for example. The studies coming from our lab are focusing mostly on splicing, because it turns out that not all Fragile X individuals are created equal. You know, some individuals do not have complete silencing mutations, and so they still do produce some mRNA, but they tend to produce a truncated version of the mRNA, like an isoform. And so Sneha's research from our lab is focused on correcting that splicing defect so that these individuals can produce full-length FMRP transcripts. So there are a lot of studies happening, and you can definitely go on the FRAXA website, and you can see what projects are being funded and also which projects are in clinical trial phases as well.
Great, thank you so much. We have some more questions in the chat. So Ivana is asking, can you speculate on a mechanism for the FMRP binding preference towards polysomes over individual ribosomes?
Yes, so this is a very interesting question because there are a lot of mRNAs that are translated by monosomes in the developing brain, in the neuropill, and we do not understand whether there's a difference in how FMRP interacts with single ribosomes or through polysomes. However, what we think is happening is there are these special population of mRNAs that are long mRNAs that encode cytoskeletal proteins important for making connections in the brain. And so these long mRNAs take a long time to translate. And so they usually are polysomal rather than monosomal. So you only need a small number of mRNA copies to be able to make a lot of protein. And so we think that this mechanism is mostly used on polysomal mRNAs and mRNAs that are targeted to RNA granules, and probably shipped and transported far away from the cell body, where you need to produce a lot of protein in a short amount of time when you get a synaptic activation stimulus. Does that answer your question?
I hope so. If not, maybe Ivana can follow up in the chat. In the meantime, we have another question from Emanuela. She says, congratulations on your great talk, Mina, beautiful. Would correcting FMRP protein after birth be enough, or is its functionality also important in fetal stages?
That's a fantastic question. Thank you, Emanuela. And basically, we see that in individuals that have a little bit of FMRP expression, those individuals tend to do better than individuals that don't have any FMRP expression. And we see this in IQ because we don't have a great metric for measuring improvements. So we can look at the number of CGG repeats and the correlation between that and how much FMRP they're making and their IQ performance. And ideally, we think that if you are an adult and we can reactivate your FMRP expression, then you will benefit from it to a certain extent. We don't know by how much. However, if you're an individual that have complete silencing, let's say, intervention at an early stage of childhood is probably the best case scenario. However, when you were talking about embryonic development, there are all kinds of ethical considerations, right? How can you consent to that? And also, where are we in terms of safety of doing these kinds of manipulations to embryos? So these are all really good conversations and discussions that we need to be having. But from what we understand, if we can restore even a little bit of FMRP, then we will see some improvement.
Great, thank you. We have another question. Is the CGG expansion also toxic by itself, independently of the reduction in levels of FMRP?
A very good question. The easy answer is I don't know, because so there are some genes, for example, where there are trinucleotide repeats that expand within the coding region of the gene. And those mRNAs can oftentimes get translated and cause these aberrant proteins that accumulate in the cell and cause a lot of destruction and a lot of problems because the cell surveillance systems may not be able to keep up with all of the bad proteins that are being made. However, in the case of FMRP, the CGG repeat is in the 5' UTR and not in the coding region. So the silencing that occurs, if you get a complete silencing, then we do not get any expression. However, if you have, let's say, the expression of a truncated transcript, like in Sneha's research, she discovered this 217 short isoform. Instead of the full-length protein, a shorter version was being produced. We still have not explored whether that mRNA gets translated into protein and accumulates in the cell. So that is something that we still don't know.
Excellent. Thank you so much, Mina. I think we've answered all questions. And if there are any more questions, please type them in the chat and maybe we can get to them at the end. So thank you so much. And we're now ready for Oscar's talk. Thank you.
Yeah, thank you, Mina. It was very informative. So with that, we'll move on our next speaker, which is Ye Duan, Oscar. You can connect to Oscar by following his Twitter account, or you can email Duan regarding his research. So Ye Duan Oscar is a co-joint postdoctoral research associate in the Ambrose Lab at UMass Chan Medical School and in Srivastava Lab at Harvard University. He has a dual Bachelor's of Science in Biotechnology and Chemistry and Master degree in Biochemistry from China. He did his PhD research in Ambrose Lab at UMass Chan by studying microRNA mechanisms. He's now studying acial worm regeneration as a postdoc. And outside the lab, Oscar enjoys playing music and cooking. Thank you, Oscar, for being our speaker today and sharing your research with us. And please take away from here.
Thank you, Ruchi, for the introduction, and thanks, Mina, for the fantastic presentation and also your introduction of the translation. Can you see my slides?
Yes.
Okay.
Yes.
Okay, so I can get started. So hi, I'm Oscar. I'm a postdoc in Ambrose Lab in UMass Chan, and also a postdoc in Srivastava Lab in Harvard. Today I'm gonna present one of my PhD projects is using C. elegans worms to study Argonaute Syndrome, a rare neurodevelopmental disorder. So firstly, what is Argonaute Syndrome? So Argonaute Syndrome is a new rare disease. The patients have mutations in the Argonaute genes, AGO1 or AGO2. And Argonaute Syndrome is firstly characterized by Emily Piton of AGO1 in 2021 and by Hans Kreienkamp in 2020 for the AGO2 spectrum. And the patients with Argonaute syndrome, usually kids, they have neurodevelopmental disorders and they exhibit intellectual disability. And some of the kids have epilepsy or hypotonia, and they mostly have motor developmental delay, and they may develop autism spectrum disorder. And here in the bottom left corner, I'm showing you a phase 2 gene facial analysis with 14 kids who have AGO syndrome and 14 kids who are healthily development. And as you can see, the kids with Argonaute syndrome do not have a very significant facial features. So and also like Argonaute syndrome is defined by the mutation of the Argonaute genes. So diagnosis of Argonaute syndrome requires genetic detection using whole genome sequence or whole exosome sequence, where the patient or the potential patient is gonna provide a little piece of the tissue, and then the scientists and the doctors will sequence your genome or your gene expression, and to detect whether you have a mutation of the Argonaute genes. By the year 2023, there are only 85 diagnosed cases worldwide, and unfortunately, most of them are in either Europe or North America. And since the diagnosis of Argonaute syndrome requires the genetic detection, there's no cases reported in any developed countries or regions with regions that do not have a robust health insurance. So the human genetics predicted that the occurrence of Argonaute syndrome is 11 cases among 100,000 children. And as I said, the Argonaute syndrome is defined by mutations in the AGO1 or AGO2 genes. So what are the AGO genes, and what do AGO genes do? And to introduce that, I will firstly go over the what is protein and what is gene expression and regulation. So firstly, thanks Mina for all the fantastic introduction, and I'll introduce the protein and the gene regulation again. So basically, protein is the core element of life. And it's very important for many activities. For example, the proteins comprises our organs and tissues. The proteins is important for us to generate energy for the activity. And also proteins help us to defend against infections or pathogens. And also proteins are important for our brains. It's not only for the function of the brains, but also for the development of the brains. And one feature for the protein is that the proteins have different levels in human. And my friends Alessia and Ruchi have a question here in the poll, like how many proteins do you think humans have? Okay, as while you are making the poll, okay, I'm interested in looking at the results. Okay, so the correct answer is over 100,000. Okay, so going back to the protein thing. So one feature for proteins is that different proteins have different levels, and the amount of proteins has to be kept in the balanced state, which we call proteome homeostasis. And for example, if you only look at those green and those blue and brown proteins, you have many blue proteins and a few brown proteins. But another feature that is important is that the protein levels can be dynamic in development. For example, as you're an infant child, you may have many blue proteins, but only a little brown proteins. But as you grow up, you may have more brown proteins, but only a little, a few blue proteins. And how is this process regulated? So to understand how regulated, let's first describe the protein. So you can think the protein as a three-dimensional structure like a knitting ball. And if you lose the knitting ball, you have a stretched yarn. And similarly, if you stretch the protein, you have a long peptide, which is comprised of amino acids. In human, we have basically 20 amino acids. And the different combination of the amino acids determines the sequence and the length of the peptide, which in turn determines the structure and the functions of proteins. And the sequence of the proteins are stored as information in the DNA, but the human cells or any eukaryotic cells cannot directly use the information stored in DNA to produce protein. So the information has to be copied in the messenger RNA or mRNA, and the cells can use the mRNA to produce proteins. And for another analogy, you can think the proteins as a house and the DNA as the designing of the designer. The builders won't be able to build a house according to the designing. So someone have to transcribe the designing into a blueprint. And then the builders can use the blueprint to build the house. So the process between the DNA and the mRNA is called transcription. And the process between the mRNA to the protein is called translation. So the next question is, how is the protein production regulated? So the protein regulation can be regulated by many, many ways. But basically, you can group them into three categories. You can change the status of the DNA and also the process where the DNA is transcribed into the mRNA. So in the case of the building, you can change the designing of the designing so that it's easier to read or hard to read. You can also give the people who transcribe the designing a sharper pen or a pen that does not have ink so that they can change the transcription. And this regulation is called transcriptional regulation. You can also change the status of the mRNA and also the rate of the translation. For example, you can change how the blueprint look like, and also you can give the builders more energy drinks so that they can work harder. And this process is called post-transcriptional regulation. And also, after the mRNA has been translated into the peptide, the protein still has to be folded and to be modified. And this process is called post-translational regulation. And the Argonaute syndrome is related to the process of the post-transcriptional regulation, whereas you manage the status of the blueprint and also the efficiency of the workers. And one mechanism that is key for the Argonaute syndrome patients is one molecule called microRNA. So microRNA is a small endogenous RNA, which is very short with only 22 nucleotide long. It is encoded also in our genome, so it looks like a gene, but it does not encode a protein. And we have many microRNAs. And microRNA is a key regulator for the post-transcriptional regulation. And it has basically two functions. One, the microRNA can destroy the blueprint, so they destabilize the messenger RNA. And also the microRNA can inhibit the translation, so to stop or slow the builders to build the house. And my colleague, Alicia and Ruchi have another question. Can you guess how many % of human genes are under the regulation of microRNA?
Can you see the poll on the screen?
Okay, so I can give the answer. So based on the computational analysis and also evolution analysis, more than 60% of human genes are regulated by microRNA. So microRNA regulation is important. Okay, so with the mechanism of microRNA regulation, we can turn down the expression of proteins during development. So then the question is, what is Argonaute gene doing in the process of microRNA regulation? So basically, Argonaute proteins are important for the production of microRNA. So you have to our cells have to use Argonaute proteins to generate the correct microRNAs and also to make the microRNAs functional. And the more important role for Argonaute gene is that the Argonaute protein will bind to the microRNA so that they can form a complex called microRNA induced silencing complex. And this complex can bind to the mRNA and then destabilize the mRNA or inhibit the translation. So in the healthy people, at some time, for some genes, there's microRNA expression, and then the target mRNA is turned down. So so you have fewer production of the protein that you want to regulate. Whereas in the Argonaute syndrome patients, their AGO genes are mutated, so their Argonaute proteins cannot be fully functional. So they can either they cannot make the correct microRNAs or cannot make correct amount of microRNAs. And also the Argonaute genes may not be able to bind microRNAs. And even they are able to bind microRNAs, they may not be able to convert the target repression, so that they have more mRNAs, which is not necessary. And also they produce way more proteins, and then they become pathogenic. So my project is to understand what is the mechanisms which differs the healthy people and the people with Argonaute syndrome patients. But I have a difficulty is that there are only a few cases of Argonaute syndrome. And also the samples from those patients are very limited. And even though I have access to their samples, there are only a few cells or some genome informations which cannot allow me to do the future experiment. So then I would like to take advantage of a model system. We call it Cynohepatitis elegans or C. elegans. So C. elegans is a nematode worm which lives in the soil and they eat the rotten apples or maybe pears or peaches. So this tiny worm have around 1,000 cells, but every cell is fully transparent. So you can see the development or the behavior of every single cell. This worm also have a very short lifespan. They can get sexually mature in 3 days, and they can live for 7 days, and they're very easy to culture in labs. And what is most important is that the C. elegans is a very good tool for easy genetic manipulation. So you can change their genomes to model the mutations that you want to study. And also many of the C. elegans genes are highly similar between humans. With humans, and especially the Argonaute genes. So for the Argonaute genes, we found that many of the mutations that we identified in the Argonaute syndrome patients are identical between human and C. elegans. So this means two things. One is these amino acids are very important because they are evolutionarily conserved between two distantly related species. And two, we can use C. elegans as a model to study the function or the impact of those mutations. So what I did in this project is I took the mutations of the human AGO1 patients, and I used a technique called CRISPR-Cas genome editing to introduce the identical mutations to the to C. elegans. And then I can monitor how the C. elegans develop. And then also I can take the gene expression pathway and sequence the mRNA, the translation, as Mina has told you, and to see what is the impact of the mutations in terms of the gene regulation. So what I found. So first thing I found is, as I have told you, the Argonaute genes are important for the biogenesis or the production of the microRNAs. And I found that in some of the AGO1 mutations in the worms, many of the expression of the microRNAs are disrupted. Here I'm showing an MA plot where the x-axis is the average expression level of the microRNA, and the y-axis is the fold change of the expression level. And the red dots indicates, each red dot indicates one single microRNA whose expression are statistically higher than normal. And the blue points indicates the microRNA whose expression level are statistically lower. So as you can see, the Argonaute syndrome mutations will cause a global disruption of microRNA production. And what is the impact of microRNA production? As we know, like the microRNAs are regulating the translation and the stability of mRNAs. So I found that in the mutant, the mRNA abundance is disrupted, and also the translational efficiency, which indicates the how your mRNA is translated into the proteins. So both the mRNA abundance and translational efficiency are disrupted. And as a consequence, the protein production is also disrupted. So basically, the patients will have a very imbalanced proteome. We also look at in terms of intact animal, what is the phenotype, or what is wrong with the worm. So we look at one specific cell type, which is called the SIM cell. The SIM cell is a stem cell, and the stem cell means that this cell have the potency to differentiate into other type of cells. So in this in C. elegans, the SIM cells, or the stem cells, the hypodermal stem cells, undergoes an asymmetry division at each molting event. The asymmetry division means one daughter cell of the stem cell will differentiate into the hypodermis, or the skin cell, whereas the other cell will remain to be stem cell. And we found that in the wild type animals, or the animals who have normal AGO genes, at the last molting events, the stem cells can all be differentiated into hypodermal cells. Whereas in the AGO mutants, at the last molting events, the stem cells cannot differentiate into the hypodermal cells, indicating that the mutations can impact the stem cell differentiation. Although the C. elegans, this is the hypodermal cells, and also C. elegans existence from human, this may indicate that the mutations may impact the differentiation of the stem cell. So it may be related to the differentiation of neuron cells. We also did some structure analysis. So here I'm showing you like a snapshot of the interior of the Argonaute protein and the microRNA. And we found that if you mutate 3 amino acid, you may have a distortion of the Argonaute protein, so that the contact between the Argonaute finger to the microRNA will be disrupted. And if you think of a knitting ball, it's like you put something in the knitting ball, and the knitting ball is disrupted and cannot be fully functional. Another thing we found interesting is that many of the genes that are perturbed in the C. elegans have homologs in human brains. And homolog means that those two proteins are probably evolved from the same ancestor or similar ancestor. And we found out that in C. elegans, many of the perturbed genes have homologs, and these homologs are related, or has been reported to be related to associate with neurodevelopmental disorder or intellectual disability. So that means using the information that we learned from C. elegans, we may have a chance to generate new diagnostic method, and also we may have a chance to develop new therapies. So in the end, I would like to thank Emily Piton and Anna Zinopieva. We collaborated heavily for this project. And also I would like to thank the members of Ambrose Lab and also Srivastava Lab. And our paper has been published a few days ago. So if you're interested, please scan this QR code. It's still functional within 7 days. You're welcome to read the paper. And also the parents of the patient kids have formed a nonprofit organization called AGO Alliance. So if you want to learn more about the information and the current therapeutic development of Argonaute syndrome, you can scan this website. And also you're welcome to reach out to me by email or follow my Twitter. And I would like to take any questions.
Great. Thank you so much, Oscar, for a great talk. So again, if you have any questions, please type them in the chat box, and I will get us started with a question. So you mentioned that you took the mutation from humans with Argonaute syndrome and you introduced it into worms. Do these worms with the mutations have any changes in their behavior in the lab? Can you see any phenotypical differences in them, or have you not done those studies yet?
Yeah, that's a perfect question. So we can answer this question in 2 steps. So firstly, we did see some worms, they have different behavior. For example, they look a little bit clumsy, and if you touch them, if you touch like a normal worm, they will like react very fast. But if you touch the worm with the mutations, they kind of respond slowly. But they definitely are different. But the caveat is that we don't know whether these differences is due to the defect in the neuron system, or this has a defect of the muscular system, because the Argonaute is a global regulator. So if you change the Argonaute protein, you mess up the whole microRNA regulating network, and also you mess up the whole protein expression system. So we don't know whether the weird phenotype or the weird behavior of the worms is attributed to the neuron defect or is other types of behaviors. And also we observed some of the targets which is predicted to be some perturbed microRNAs, and also their reported phenotypes are also different. For example, if you in the mutant of the Argonaute syndrome, some worms may have some defects in tasting the high salt. So they cannot respond to the high salt, which is normal for worms. So yes, they do have some behavior they do have some difference in the behavior.
Got it. Thank you. That's very interesting. Another question I had is, you showed the graph where you disrupted AGO1 in the worms and some microRNAs went up and some went down in expression. So my question was, yeah, the one on the left, does that mean that AGO1 negatively regulates some microRNAs and it positively regulates others, as opposed to globally being involved in microRNA expression?
Oh yeah, that's a very, very good question. So the direct answer is we still don't know, because the expression of the microRNA is related to many factors. It's both from the process of the DNA to the precursor to the primary transcript of microRNA, and also the processing of the microRNAs. And we know many of the genes that is important for the processing of the microRNAs are also regulated by microRNAs themselves. So that means the mutations of the AGO genes have 2 impacts. One is in their real role to regulate the microRNA expression, they may have some defects. And also, if we have some change of the, like, let's say the primary microRNAs, or the first batch of microRNAs, these microRNAs may in turn have some defects of the targets, which are also involved in the microRNA biogenesis pathway. So yes, so the answer to you is yes, the AGO syndrome mutations do have a great impact of the microRNA biogenesis, but what is the detailed mechanism? We still don't have a clear answer. We just know the consequence.
Got it. Thank you so much. If anyone else has a question, please type it in the chat box. And if not, I will hand it over back to Ruchi. Thank you, Oscar.
Thank you.
Thank you, Oscar. So with this, we will so the Science Cafe will be done. We would like to thank you, our speakers, once again, Meena and Oscar. And I'm Ruchi Sharma, along with Alicia and Oscar. We organize Science Cafe. And if you are interested to join our team, you can email us at umass.edu, or you can be a speaker in Science Cafe and share your research with the community. You can also follow us on our social media handles on Facebook, Twitter, and Instagram. With that, thank you very much for your attention and time. Thanks.
And please take the survey that I posted in the chat. Thank you so much.